I'm going to talk about my PhD project, which was focused on Bifidobacteria and how I use genomics to predict their carbohydrate utilization capabilities. So many of you probably have heard about Bifidobacteria. So these are gram-positive microbes that are commonly found in the human gut, especially in infants. And the important thing here is that most of the species are generally considered beneficial for the host, mainly because they secrete a wide array of metabolites, beneficial metabolites. And this is one of the reasons why many bifidobacterial strains are used as probiotics, and sometimes in synbiotic formulations where probiotics are additionally supplemented with some kind of glycans. And what I want to emphasize here is that bifidobacteria are really saccharolytic microbes. So they really like to eat sugars. And if you look at their genomes, previous estimates suggest that approximately 10% of their genomes are dedicated to carbohydrate metabolism. So it's important to study this metabolic process. And in my work, I wanted to answer two questions. First, we do know that different bifidobacterial species and subspecies prefer different glycans, and it's often associated with different ecological niches of these species. But I was curious, what is the full spectrum of these different metabolic pathways that differentiate bifidobacterial species? And also, numerous studies have shown that bifidobacteria vary at the level of strains. So strain 1 can metabolize a specific sugar that strain 2 cannot metabolize. But I was curious how the strains isolated from some underrepresented populations contribute to this strain level diversity. And the main technology that I used in my work was essentially genomics-based metabolic reconstruction. So the idea of this method is pretty simple. So metabolic reconstruction is similar to putting together jigsaw puzzles, but in our case, the puzzle pieces are these functional elements of metabolic pathways. For example, glycan transporters that uptake various glycans into the cell. Then glycoside hydrolyzers. These are very important catabolic enzymes that essentially break down complex glycans like oligosaccharides to simpler ones. We also look at the so-called downstream catabolic enzymes, and also we look at transcriptional regulators that control the expression of genes encoding all these different pathway elements. So the main idea of metabolic reconstruction is look at genes encoded in genomes, look at genes in genomes that encode all these different pathway elements. And then you can look at strain 1, for example. If its genome encodes a complete metabolic pathway, we say strain 1 is a putative utilizer, in this case of xylotriose. Whereas strain 1 might not encode a transporter for xylotriose and a glycoside hydrolase, which is important for degrading this oligosaccharide. In this case, we say it's a putative non-utilizer of xylotriose, and I assign a binary phenotype 0. When we do this metabolic reconstruction, what's very important is to know what genes we need to search in the genomes. And the problem here is that a lot of this information is not represented in public databases. So what you have to do is go into primary literature and step by step dig this information about these various glycoside hydrolases and transporters, and then propagate this information on various genomes and strains. But in addition to kind of capturing what is already known, we also can predict novel metabolic functions. And the main idea here is the idea of co-regulation. Basically, genes that are co-regulated often encode proteins that are involved in the same metabolic pathway. And this is one of the approaches how you can predict the specificity of sugar transporters, especially if they co-regulate, for example, with glycoside hydrolases with non-function. And here I just want to demonstrate how the manual curation compares to widely used tools like PROCA or eggnog mapper. So here I manually created approximately 40,000 annotations in 263 reference genomes. And as you can see here, this manual curation considerably improves the quality of functional annotations, especially for transporters. So for transporters, over 90% of annotations were improved. And this, I think, is important take-home message from this talk, is that when you use this widely available tools, be aware that the standard annotations for transporters are not very good, so you have to additionally manually create them. Now, going back to this whole metabolic reconstruction approach, so initially my work was completely manual, so I reconstructed 68 carbohydrate utilization pathways in these reference genomes, but then we used this database to build a tool called GlycaBif, which allows you to analyze the representation of these 68 reconstructed pathways in any number of genomes. But additional important thing when you do this genomic predictions is, of course, to test the predictions. Are they good or not? To do that, we used various approaches, but most of them essentially were like growing these different bifidobacterial strains on different carbon sources. But overall, we achieved pretty good prediction accuracy. So it was over 90%, which is I consider pretty good. Now I want to show some of the results of my work. So here is a heat map that shows you the representation of these 68 reconstructed pathways in approximately 3,000 Bifidobacterium genomes. And as you can see here, there's a lot of variability. So different species and subspecies, they harbor distinct sets of metabolic pathways, which again reflects distinct ecological niches of different species and subspecies. For example, infant gut versus adult gut. But also, there's a lot of variability at the level of strain. Essentially, many pathways are not conserved within a species or subspecies. And it tells us that at the level of strain, there can be a lot of metabolic tuning. Essentially, some strains can do way more than other strains, but still belong to the same species. And kind of going into more details about that, I want to talk about the Bifidobacterium longum species. This species is very well known because it's you can find information about this species in many studies focused on infant gut. But unfortunately, this species has a very complex subspecies structure. Here I'm showing a heat map which shows average nucleotide density of genomes I selected. But basically, there is subspecies infantis, there is subspecies longum. Here I want to emphasize this novel clade we found, which is kind of similar to subspecies longum, but yet slightly distinct based on average nucleotide density. But when we looked at the representation of these reconstructed carbohydrate utilization phenotypes, we found out that this clade, which we call BL-NOV, has significantly lower number of reconstructed pathways. And specifically, it seems that this clade has lost a lot of pathways involved in host glycan utilization, in this case, human milk oligosaccharides, but instead it gained the ability to metabolize certain plant polysaccharides, such as soluble starch and pullulan. And again, we validated these predictions using in vitro growth experiments. And overall, this tells us this novel clade within Bifidobacterium longum, potentially subspecies, likely has a distinct ecological niche compared to subspecies longum or subspecies infantis. Going further, I want to kind of do a small refresh about human milk oligosaccharides. I already mentioned them, but basically these are glycans found in human milk, and there are many different structures, over 100 of different structures. But you can group them into neutral HMOs, neutral fucosylated, and sialylated based on these different types of modifications. What's important, most of these structures cannot be metabolized by our body, but they can be selectively metabolized by Bifidobacteria. And over the years, multiple works have been dedicated to studying this HMO metabolism by Bifidobacteria. For example, many gene clusters have been implicated. One of them is called H1. Initially, it was discovered in Belongos species infantis, but in our work, we were able to identify orthologs of this gene cluster in Belongos species and Bifidobacterium cassionahensis strains of Bangladeshi origin. So these strains were isolated in Jeff Gordon's lab at Washington University. But what's more interesting is that some other Bangladeshi strains, again, cassionahensis and Sioux, they harbor a slightly distinct gene cluster, which I called FHMO. And subtle differences between these clusters involve transporters. For example, the H1 cluster encodes multiple transporters. We don't really know their subset specificity, but FHMO cluster encodes a different transporter for fucosylated HMOs, which was very well characterized. So we next decided to look how this difference in gene content kind of translate into phenotypic differences. We collaborated with Lars Bod lab at UC San Diego, and found out that these strains with H1 clusters here in green, they essentially grow the best in the medium supplemented with HMOs. And they can metabolize all these distinct HMO structures, but they slightly struggle with short fucosylated HMOs like 2'-fucosyl lactose. Whereas the strains with the FHMO cluster, they do prefer these short fucosylated HMOs. And if we kind of combine the growth and HMO utilization data together, you can see using principal component analysis, you can see here that strains essentially cluster by the presence of this specific gene cluster rather by taxonomy. And this tells us that it's really important to look at the subtle gene differences, especially when we're talking about genes encoding HMO transporters, because they essentially dictate how different strains will utilize different HMOs. In conclusion, I actually want to talk about one of the limitations of this work. Although we did study a lot of strains isolation for Bangladeshi children, a lot of our genomes still were mostly from westernized populations. And I think in the future, when more datasets will be available, we will find a lot of novel information about novel metabolic pathways found in these bifidobacterial strains isolated from underrepresented populations. And generally, I think we do need more experimental work on glycan transporters and in kind of including them into metabolic reconstructions. So with that, I'm gonna conclude my talk, and I want to thank the collaborators who were involved, especially Jeff Gordon's lab at Washington University. And if you're interested in details, we recently published this manuscript, also a tool for predicting the pathways in bifidobacterial genomes. So thank you for your attention.